This is what it looks like. So you hear the crunch, 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 the other saying, how this one, and then you video. It just said on Thursday. So, yes.
Okay, so we're live, which means that
So you hear the same. It's cold.
yeah, it did get cold. So glad that we didn't get any of the snow that they have in Western New York, though.
Oh my gosh. Yeah.
Yeah. How are you doing?
Good.
I'm going to attempt to broadcast this live to folks who can't come. Come on.
Yes, right.
Via a YouTube live broadcast.
Oh, really?
Mm-hmm. Come on. Yeah. How was Mexico?
It was really good.
Oh, wonderful.
A long week. A full week. So that my talk went really well. And met lots of people. New collaboration.
Nice.
People from all over Latin America there. Cool. How was the work there? It was work. Thank you so much.
Yes, perfect. Thank you, John.
Do you need anything else? No, I think I'm good. Okay. Wait, what is this?
Oh, that is a design that I did for the subterranean street biology meeting.
Oh my goodness.
It's just my background.
Whales and is that a whale?
No, that's an Alabama cavefish.
Oh,
and,
really?
Ozark big-eared bat.
Oh my goodness. How interesting. Do you know Tom Illiff?
Who?
Tom Illiff?
No.
He works on yellow peas and
Oh, cool.
things that it does a lot of cave diving and we're developing new rebreather techniques and so. So it's pleasant. But that's good. Have you been there? Yeah, that's George, that's Sergio.
Just kidding.
That was for the, Huh? What's wrong? Get the eyes? Yeah, exactly. What else is new? So when were you going? Yeah, because every time I go to yeah, no, I see her. Yeah, that's the whole thing. But I should tell you, we need a partner, so they took another. And I don't want to fish. Well, I don't know about fish. Trevor, can you do like, well, we'll just see if we get to know. Phil, does that 103 years affect the? Got your light on.
So that's the source. That's
Yes, that is the source.
There's a whole parade coming from.
Yeah, we're coming.
That's the hope.
Oh, that's my favorite time of the
That's your drink there.
This is my put away. Is your mother still writing names?
Okay, so you got all the people in my lab.
Oh, yeah.
Okay, I think we're going to try to start more or less on time. So Kate can tell you everything she's been up to. And I'm introducing Kate, who's giving you her thesis seminar today. So I first became aware of Kate. Oh, when she sent me an email from I got an email from this young lady who is at a department at a small school in New York State who have been working on bat vision. And I thought, well, anybody who works on vision in real animals is somebody who belongs in my lab. So I invited her down for a visit. And it took me probably a few weeks before I realized just how appropriate it was that Kate had chosen bats as something to study. It sort of fit with what I discovered about Kate as she came up to the lab and started showing her all the sides of her personality, which really made the lab a much more interesting place the last few years. We are going to miss Kate quite a bit. And people are going to still hear me walk into the lab, I'm sure, for the next several months and say, Kate! At the top of my lungs as I walk in. And there won't be any answer from that back corner where I can't see her from the door. It'll be pretty sad. She's brought a lot of life to the lab, a lot of personality, a lot of talent, a lot of really good ideas, a lot of great research, and some really good times both here in Baltimore and with the students. So I'm going to tell you a little bit about her work. This kind of, I think, really is a picture of her dealing with her thesis. The story actually has a funny sort of coda because I actually took this picture and I said, wow, a picture of Kate with her thesis, and some random hiker who's walking by burst into laughter. So I guess it's one of the ones that's on this hill. So she eventually got on top of the rock, but you can see she's kind of tenuous up there still. You know, all who tell the thesis know that grabbing a hold of it's one thing, getting on top of it's something else again. But eventually, everything settled down. She got up there and she drank up some fun with her. So everything worked out okay. I took this picture up and realized as I was going to show it that not a single person in that picture actually resides in the continent of the United States, which is kind of surprising. So she did manage to finish, and she's going to tell you about her research today. So here's Kate Feller to tell you about her PhD.
Okay, well, thank you so much for all of you to come out for my dissertation defense. If you're not aware, I'm also broadcasting it live on the YouTubes for those who did not attend. So mind your conversation, it's live. But today I'm going to be telling you about the visual ecology of stomatopod larvae. I never use the clicker, so bear with me. Okay. Oh, turn it on. To those of you not familiar with the field of visual ecology is the study of animal visual systems, but in the context of their environment, as well as their ecology. And so this has to do with the visual system and how it actually works, so what kind of light that it's has evolved to detect, the physiology of the visual system, the visually mediated behaviors of the animal. This also involves how an animal can be perceived by a viewer. So predator-prey interactions, camouflage, those are all considered visual ecology. Pretty diverse interdisciplinary field. And so I use the stomatopod larval system in order to examine some questions in visual ecology. And so as an outline, just of what I'm going to go over in today's talk, first, I'm going to give you a bit of background on exactly what a stomatopod is, some information about their ecology, their life history, as well as how we identify them. And then I'll get into a story about larval vision in these remarkable animals. A different story at the other end of visual ecology about eye camouflage, and then a quick summary and acknowledgements. So, starting with stomatopods. So stomatopods are an order of malacostracan crustaceans, and all that means is that they are a big, that is a big classification for a group of crustaceans that's totally different from anything you order at Red Lobster. Anything you order at Red Lobster is a decapod. Stomatopods, there's over 400 species. Stomatopoda is the order. They're doing their own thing in terms of evolutionary history. It's a very diverse order of crustaceans with those 400 species. They're located globally in all of the world's oceans. They're strictly marine. They come in a variety of different colors and sizes and shapes, and they're really remarkable little animals. Their common name is the mantis shrimp because much like a praying mantis, they have a large pair of hunting appendages that they hold out in front, and they either smash prey or they spear it as it swims by. You can see that pretty well in this little picture up here of that hunting appendage. So as far as adult stomatopods go, they occupy, in that global distribution of them, they occupy a very broad diversity of depths. So they live in very diverse marine habitats from the intertidal zone that's very shallow here on this graph, all the way down to thousands of meters. Which, if you go along this depth gradient in the ocean, that encompasses some very diverse photic environments. And the light environment changes quite drastically as you go from near the surface to the depth. And within the order of Stomatopoda, you have 9 superfamilies that you can then further divide them into, which they're color-coded with this list here, with where they're found at the depth. And I starred the 3 major ones that will be covered in my talk. These, the Gonodactyloids, Lasioscoles, and Sculoids, the majority of species that at least we capture at our field site fall into these 3 superfamilies. Stomatopod biodiversity is greatest in areas of coral reef systems. And so this contour map demonstrates in the Western Pacific and Indian Ocean, so we have Australia here, regions of species where the number of species are found. So you can see that in this area around Indonesia, you can have as many as 45 or more species. And I highlight this particular region of the world because this is where the majority of my field work is conducted at Lizard Island Research Station, which is here in the Northern Great Barrier Reef, where you can see within the orange and red high biodiversity regions. And currently there are 32 described adult species found at this location. Stomatopod crustaceans go through a pretty typical life history or developmental cycle of other marine crustaceans, where you have your adults. This is a male just poking his eyes out of his burrow. The adults live on the bottom of the ocean, or the benthos, in burrows made out of rock or sand or coral. The adults then find each other, mate, lay a clutch of eggs that then hatch into larvae that then leave the burrow and migrate up into the open ocean, which is also known as the pelagic environment. And so larvae will then occupy the pelagic environment for a series of larval stages that can be anywhere from as few as 3 stages to as many as 11. It's very hard to characterize larval stages because they're difficult to culture in the lab. So most species, we don't know how many larval stages they have. But they progress through these stages. Each stage is separated by a molt where they pop out as the next stage. And they swim around in the open ocean, feeding and avoiding being eaten, and looking for the adult habitat. When they reach their terminal larval stage, they then molt into a pulse larva, here, and that pulse larva then begins to resemble the adult and migrates back to the benthos, where it will then occupy its adult years. So one of the major challenges of studying stomatopod larvae, as well as many crustacean larvae, is that they don't look anything like the adults. And so the adults are what all of the species descriptions have been based off of. So if you have a species that has been described at Lizard Island, for instance, you can almost guarantee that there is no description of any of the larval stages. And so it poses this great challenge of how you can identify an animal that you capture in the wild and tie it back to that species. And these are just some photographs of different species of larvae. You can see that they are they're almost very similar in some of their morphologies, and except they're also different in terms of, so this is actually one species, these two right here, that actually look quite different, though they are transparent and have shiny blue-green eyes. And so tying them back to a single species can be difficult if you only have morphological identifiers and it hasn't been described before. And so the method that I have used throughout my entire dissertation in order to get around this identification issue is called DNA barcoding. DNA barcoding is where you use a DNA fingerprint or barcode to tie an individual back to its identified species. And so the sort of accepted DNA fingerprint for crustaceans, at least, is the cytochrome oxidase 1 mitochondrial gene, which this is a picture of the species Harpioscola harpax and its mitochondrial genome, which has been sequenced. And so that gene is located right there on the genome map. And so the way that DNA barcoding works is that you sequence the CO1 gene from an individual that you have no idea what it is, and then you compare it to a database or a lot of known sequences that you've collected from adults. And so when you do that comparison, if the sequence in question is less than 3% different, or has a less than 3% sequence divergence from an identified sequence or reference sequences, then you can say with certain confidence that that unknown is identified to its respective species. And so this is a phylogenetic tree that I generated with all of the CO1 barcodes that I collected throughout my dissertation. There's over 300 here that are all highlighted in either green or red. And they are compared to, I think I have 67 adult barcodes. So that's 67 out of 400. We still have a lot of work to go in terms of generating reference sequences. But you can see with these results, so 20 of those reference sequences are species from Lizard Island, where we know there are 32 adult species. And so, since most of these sequences were collected from individuals found at Lizard Island, you can see that I have 20 identified species within this clade. And so everywhere that you see green is where I have an identified species. And just a point of note, where you see these wide triangles, that just means that I have a lot of reference sequence or of larval sequences in it. So that just means that there's a lot there with 1 or 2 reference sequences of that species. Interestingly, from these findings is that I have 26 unidentified species. So if you do a little math, 32 minus 20, that's 12 sequences that were missing of the known species at Lizard Island. However, I've found 26 that we don't have references for. So that means that there is more species diversity going on at Lizard Island than we observe in the adults we find there. And that could be related to actually species not being described for that region as adults. It could be because I captured individuals that were not normally, that are not supposed to be there, that they got off track in their larval dispersal. We can't really tell until we have more references. As a side line of research to my main body of work, I've also begun producing larval descriptions of the morphology of some of the common species I find at Lizard Island. And so this is an illustration that I did that was published in 2015, since the first piece is cut off with it, where we tied the molecular barcodes and the morphological characters of these 2 Alema species to their adults. So if you all go to Lizard Island someday, you can use that paper as a guide in order to identify those 2 species while you're there. And so with that, we're going to move on to a story about larval vision. 2 studies that I've done on the photoreceptor sensitivity in larval eyes, as well as the physiology of eye development. So just a little bit of background. Stomatopod eyes are crazy. That is the adult's eye, the adult eye's eye. So stomatopods have compound eyes, which you can see right here. This is a beautiful adult compound eye. And a compound eye is composed of hundreds to thousands of, or just a few, units called ommatidia. And so an ommatidia, or ommatidium, can be viewed on the surface of the eye as these facets. So that is the end of each ommatidium. But if you look at it in cross-section, the ommatidia are these long units here that are turned on their side. So if you actually cut this eye like this, turn it on its side, this is what it looks like in a live tissue. And this is a schematic of what we're looking at here, where you have 2 general layers. You've got a light focusing layer, where you have the lens and the crystalline cone, and light is focused through this onto a photoreceptive layer. In adult stomatopod eyes, there is a great diversity of photoreceptors, photoreceptor classes found in the eye, signified by all of these different colored curves that correspond to the different colors in the schematic. Most of the photoreceptor diversity, such as depicted here, is in this unusual region of the adult eye known as the midband. The midband is a row of between 2 and 6 enlarged ommatidial facets, as signified here and then blown up here. And in the elaborate 6 midband row is where you have this expansion of photoreceptor diversity. So just to ground you with in something that you're more familiar with, I'll compare this to a human visual system, where we see a pretty broad range of colors or wavelengths of light, but we do that using 3 photoreceptor classes, a blue, a green, and a red, whereas the stomatopods use a lot more, up to 16. So the way that these, so a little bit of anatomy about these ommatidial units. So as I said before, it's this long structure that is almost like, if you imagine the eye to be like a bouquet of straws, this is how a compound eye is arranged, where you have your crystalline cone and your optics out here, and then that photoreceptive layer. In the adult stomatopod eye, the optics are such that light from only a single ommatidial facet is focused onto a single photoreceptor beneath it. And that photoreceptive unit is called a rhabdome, which is this structure right here. And so any light coming from off-axis or from neighboring ommatidia is either absorbed by screening pigments that are in between the optics, or by screening pigments that surround the rhabdome within the retinular cells that protrude microvilli in order to create the rhabdome. And so in this photograph, you can just see a stomatopod. This is actually a 2 midband row species from the side that's backlit, and you can see it has these very short optics, and then the retina is visible beneath it. Larvae, by comparison, also use a form of apposition type optics, where light from a single ommatidial facet is focused onto a single photoreceptor. Except in larvae, the crystalline cone region of the eye has been extremely elongated. So the reason for that is so that they can have a more transparent eye. So these are called transparent apposition type optics. And what's really interesting about this is there's an optical effect where you have a gradient of refractive index that serves to take off-axis light and focus it away from your photoreceptor and onto that light-absorbing part of the retina. And so the eye does not need those screening pigments in between the ommatidia. So these are some photographs of these eyes where you can see they have these very long optical units surrounding an opaque retina at the center. And we'll get back to a discussion about why they might want to have a transparent type eye a little bit later. So in looking at these magnifications of larval eyes, you can notice that there is something pretty different between them and the adult eye, namely that there is no midband. It's a uniform array of ommatidia. And so larvae, unlike adults, are only known to express a single class of photoreceptor. And so these are data from a study that I performed using a technique known as microspectrophotometry, or MSP, where you're able to shine light through a single photoreceptive unit or a single rhabdome, and you're able to measure the wavelengths of light that are absorbed in that photoreceptor. And so in green here and in blue are those raw data of the light that's been absorbed, and they're fit to a visual pigment template, which is in white. And the peak of that template is what we refer to as the lambda max value, and that's how we talk about photoreceptors. And so the peak wavelength of absorption in this species, Lysosphena maculata, is 501 nanometers, whereas this species, Gonodactylus finis, is 445 nanometers. What was really interesting in this study was that both of these species are known to occur in the same location. So they're occurring in the same or sympatric habitat of open water with the same spectral properties, performing similar visual tasks. tests, and yet we unexpectedly found this significant difference in their visual pigment class. And we only found a single visual pigment class in each of these species. I actually examined a total of 8 species, greatly expanding our data set on larval visual pigment diversity. And so you can see highlighted in dark are the species that I characterized using MSP versus previous publications. And you can see that my findings reinforce that larvae only have a single class of visual pigment, or photoreceptor, not visual pigment. We only have a single class of photoreceptor, whereas in the adults, you can see that they have the greater diversity in species with the 6 midband row. In adult species with just the 2 midband row, there is only a single, there's also only a single class of photoreceptor known because it is a reduced or degenerated type eye. So the findings of this study beg the question, since larval eyes are so different from the adult eyes, how do you transition from a larval eye into this complex adult structure? And so what a lot of decapod crustaceans do, because the decapods, they also have a larval phase that doesn't look anything like the adult. And the larval phase, when it undergoes its metamorphosis, modifies that larval eye, that larval compound eye, and just makes the new adult structures, even if they're more complex than the larvae. Stomatopods, however, are different. Stomatopods undergo a type of eye development that we refer to as the double retina eye development. And so this is a sequence of a single individual in its last stage. You can see that it has this larval single retina, and you can just make out a little shadow of the adult structures that are going to come in completely separate. So the adult structures, just prior to metamorphosis, develop adjacent to but separate from the existing larval structures. And eventually the adult structures take over the eye as the larval ones degenerate. And so you can see by day 1, you have this reddish adult retina coming in each eye. By day 2, the adult retina is much more well developed and has its own pigments. And by day 5, that larval retina is only a small remnant near the lateral margins of the eye. And so I wanted to know what's going on here. How do you see with how are these animals seeing with 2 retinas, one on its way in and one on its way out? And so I decided to investigate that in a species, Stomatopod, which is actually a Western Atlantic species that is fairly easily accessible at the Duke University Marine Laboratory down in North Carolina. This species was very nice to work with because the adult has the reduced 2 midband row eye, and the adult only has a single photoreceptor class in its eye that happens to be almost identical to the larva. So these squiggly lines here are the larval and adult visual pigment curves. So we can use a single light stimulus to stimulate each of these retinas and not worry that they would have a different response if we wanted to measure their physiological responses. And so in previous studies or my previous work with doing the MSP, I found that when I performed MSP on a double retina such as this, where you have the adult retina is fairly well formed, that I was able to get from a single prep terrific larval absorption data and almost nothing from the adult retina. So that led me to hypothesize that perhaps something's going on with the development of the adult retina and it's not physiologically active until post-metamorphosis or something like that. So I decided to use electrophysiology to investigate an ontogenetic series of eye development in Stomatopod. So Stomatopod is great because it's really accessible in the Western Atlantic Ocean. It has the single visual or photoreceptor class in both the larvae and the adults. And also, all of the morphological stages of Scalimfusa have been described. And so I'm able to capture and identify just in my hand without using DNA barcoding the 8th larval stage, which only has a single retina for the duration of its stage. I'm able to capture the terminal stage, or the 9th stage, which will have a single retina at the beginning, and then will form a double retina towards the end of this stage, just before it metamorphoses. And then post-larvae, which will have presumably the double retina. So I used a technique called electroretinography, or ERG, which is an extracellular reporting method where you take a microelectrode and you stick it into a tissue of interest, in this case, the adult retina as it emerges, or the larval retina as it's degenerating in this double retina preparation, and we perform single and or dual channel recording. So that just means we are able to record from a single retina or from both simultaneously. So what we do is we put this microelectrode into the animal, which would be found here under this microscope, in the schematic with the microelectrode. We deliver a light stimulus to those retinas, which then would elicit a voltage response from the eyes that would be detected and amplified by this system here, and then we get a readout on the computer. Those type of data look like this. This is the voltage response to our light stimuli. We presented 0.75 second stimuli at different log irradiances, so just at different amounts of light that change on a log scale. And so if you deliver the stimulus and you give a bright light, it's a bright light level, then you get this very strong deflection in the voltage curve, whereas you get less of a deflection as the light increases. And so this is called a voltage. So this is a voltage log irradiance response that we generated. So this is microvolts in this scale. So from these data, we then can generate a what's called a voltage log intensity curve, or V log I curve. And all that is in these data are those stimuli that we delivered plotted against the voltage response that we elicited from that stimulus. And so this is for each one of those different larval stages. And something you can see right off the bat is we get a robust response from the adult retina in the double retina, even at its earliest emergence. So when it's just a shadow, as we saw in this picture here, so it's clearly not very developed in this, we still get a great V log I curve out of it, which is the dark green curve. So whatever was going on with MSP was not correct. This thing is alive and well and working. So if we want to perform any kind of statistical comparison on the responses that we found, we have to get an actual number in order to compare. And so that value is called the log K value. And all that is the value of the log irradiance at which you elicit 50% of the response on this curve. And actually, I should point out that the points here are the actual data, and the lines here are a model that have been fit to the data, so that we can then calculate that log K value. So then if we plot the log K values against one another with, we can see from the Z8 to the post-larva, same color scheme as we have here. These are all of the log K values generated from every retina that we investigated. We can then perform those statistics and determine that, in fact, during the double retina period, the eye is more sensitive to light than either during just the larval retina, where it's a single larval retina, or where you have a single adult retina in the post-larval stage. We also found that there are changes in the response dynamics with ontogeny. So in the post-larval retina, we have the the window of light stimuli is smaller in order to go from your min response to your max response, which generates a steeper curve, which you can see here. And if we, which is signified by slope, which is what we're looking at here, then in the single retina eyes. Whereas the double retina eye has an intermediate response, presumably because it has both retinas in the eye. So we wondered so one of the things that we observed in doing this study is that Squilla and pusa undergoes an insanely fast eye development. So originally I showed you a schematic of an individual Poiosquilla thomasini from Lizard Island that takes 5 days to go through its double retina period. Squilla and pusa does its entire double retina progression in about 12 hours, which is highly unexpected. And so that forced us to do these recordings at all hours of the day. It is well known in the literature in other crustaceans and in previous studies of Squilla and pusa adults that there are cycles in sensitivity throughout the time of day. And so we needed to look at if these effects could be confounding our results. And in the present data set, they do not seem to be. There is a trend, if we just look at those log K values from what I presented earlier, just for the Z9 single retina, and you look at the ones that were measured during the day and the ones that were measured during the night, while there is a trend where the eyes are more sensitive at night, there is a trend towards that. This trend is not significant. And so there's the potential for circadian cycles and sensitivity, but we didn't see it in this study. So with that, the future directions for studying larval vision, we need to follow up with the ontogeny of MSP. So actually, I forgot to mention. With the diversity, so we found an unexpected diversity of visual pigments in sympatric species. And there's sort of circumstantial evidence that that may be related to larval stage. And so early stage larvae seem to have a very short wavelength shifted photoreceptor class, whereas the late stage seem to have a longer shifted photoreceptor class. And so a specific study that looks at MSP of a single species in early stage larvae versus late stage larvae would resolve that question and also probably help explain why we're seeing this kind of diversity in the same habitat. These data are also useful for molecular studies of visual pigment proteins or opsins in larvae and throughout their ontogeny. With the double retina eye physiology, or ERG, definitely we need to follow up with the circadian cycles and larval retina sensitivity and do a proper circadian experiment where we just measure how a larval retina changes in its sensitivity over a 24-hour period, as well as follow up with spectral sensitivity response changes to the eye and the critical superfusion threshold, which is related to changes in motion vision sensitivity. These are common things that are typically investigated with ERG and would be interesting to add to this body of work. Something I would very much like to follow up on in my postdoctoral post-larval phase is to look at the double retina neuroanatomy. And so in this double retina eye, it's very obvious that we have two retinas. But it is less obvious how the brain or the visual processing structures behind these retinas are also changing at metamorphosis and becoming rewired. So this is just a schematic of a hypothesis I have that shows those neuropiles. There's 4 of them, and that you only get partial duplication of those as well during metamorphosis. So with that, I'd like to bring you back to our basic understanding of visual ecology. So I've just told you a story about larval eyes and how they function, and a bit about their physiology and how they develop. Now I'd like to change gears and talk about larval camouflage, specifically eye camouflage, and tell you a little bit about its contrast spectrum and structure. So, larvae live in the open ocean. There's no place to hide in the open ocean. You can't duck behind a rock if you see a predator coming. And so one of the ways that larvae have evolved to avoid being eaten by other things is to be completely transparent. So here on the left, we have a photograph of an animal that's been imaged in such a way so you can see it. If I didn't image this so you could see its glass-like body, and you were just looking at it in a bowl of water, the only thing you would see are these two little black dots, which are their compound eyes. And the reason for that is that eyes always have to have an opaque screening pigment. You need to have screening pigments in between your photoreceptors so that they don't get light contamination from their neighbors. You can preserve that spatial resolution of the world around them. So while they've evolved those transparent A-position optics I described earlier, they cannot, so they're as transparent as they can get, it can't be completely transparent. And this is a nice photograph just showing how transparent these animals can be when not imaged in such a way. This is that photograph of an adult male poking his eyes out of the burrow, and swimming around him are about 40 of his progeny that are fairly transparent. You see that, and the only thing that's really visible is there's two little black dots that are their eyeballs. And so, a feature of all stomatopod larval eyes examined to date is a structure, a light-reflecting material referred to as eye shine. Eye shine has been observed, like I said, in all species that have been examined, as well as throughout the entire larval phase of a given species. So in this particular species, in the embryos, you can see the eye shine coming in as they approach their later development. And then in the earliest larval stage, you have this really bright blue-green reflecting material over the retina, as well as in the late stage where you have the adult retina coming in for that double retina. So our hypothesis was that eye shine functions as a camouflage in the pelagic environment. So this is how they are reducing the contrast of that opaque retina that they just can't get rid of. And that because it's very blue or green, depending on the species you're looking at, that perhaps the spectrum that is reflected off of this eye shine is matched to the background to further reduce the contrast. So you might be tempted to think that eye shine has some sort of visual role with the animal. However, that is immediately discounted by the presence of what we call a pseudopupil. So pseudopupil is the region of the eye where you have the ommatidia are looking directly at the viewer. And so they appear black in this adult retina. So these are the ommatidia that are looking directly at the camera. And we see that pseudopupil in the larval retina. So you can see it here, everywhere there's an arrow, you can see a pseudopupil, even if it's only a few ommatidia big. So it's unlikely that eye shine influences the animal's vision. We also, just based on looking at the gross observation of the eye, that this eye shine material, while absent from the visual pathway, which going through the optics down to the photoreceptor is the visual pathway, that it lies at this junction between the optical layer and the photoreceptive layer. And that's where we'll find this material. So the first step in testing this hypothesis was to photograph larvae in their natural environment and measure their contrast with that natural lighting. And so the way I did this was I captured a bunch of larvae, I glued them to sticks, I mounted them in front of a camera, from here and here. Then I took them down on scuba, mounted them on a tripod, and took photographs of them in a variety of different depths and a variety of different directions relative to where the sun was coming from at different angles to the horizon, as well as different rotations of the larva looking at you in different directions. I then calculated a value known as Michelson contrast in order to determine the value of contrast from those images. And all of this is a calculation that takes the luminance of an object, in this case the light that's coming off of our eye spot, you subtract that from the background, and then you divide it by a summation of those two variables. And so you get a scale of numbers that run between +1, where the object is much brighter than the background or whiter than the background, and -1, where it's much darker than the background. And this is just a video showing you what all these different values look like. And at the value of 0, you have no contrast, so the two are equal. This is a really a nice way to talk about contrast, because we can then go back to this grayscale sort of imaging for communicating. And so these are what the actual photographs that I extracted the data from look like, where I measured a 15-degree circle around my larva of interest, and I averaged the pixel value, the green pixel value, and the RGB information from the image. I measured the average green pixel value for the luminance of the background, and then I measured the average green pixel value of the eye spot, which is depicted here in our blown-up animal from that same photograph. Also imaged in every data image was a black standard, so that we would know what a dark object looks like on the natural background. So we calculated that Michelson contrast value, and then we had a huge data set then to look at for all of those different angles and directions and rotations. And so this is just a plot of all of those Michelson contrast values depicted on a grayscale that corresponds to each value. So each point in the plot represents a different rotation of the animal, whether it's looking at you with its ventral surface or its dorsal. I also imaged the animal straight up and down in the vertical, or 90 degrees horizontal. So that's why there's 2 rows. The last 2 dots in every pair is that black standard and what that looks like on the background. And then in each ring, we have the different angles relative to the horizon, and in each direction relative to the sun. So the take-home message of this very complicated slide is that eye shine is always less contrasty than the background than a dark standard. And so if we look at all of the data generated from these thousands of images, you can see that in fact the eye shine contrast values are much closer to this value of 0, no contrast, than any of the black standards that we measured for any lighting condition at any depth, any direction. If we break this down into a box plot, just looking at the distribution of data with the horizontal lines of the median, the interquartile ranges, and we break it down by each individual that was measured from 3 different species at different times of day with different elevations of the sun and at different depths, you can see that there really, none of those variables seem to affect eye shine. It's always doing a good job. As a secondary, it's always doing a much better job at least than the dark standard. I also had the opportunity to image a dead animal, because it turns out when larvae die, the eye shine is reduced. It doesn't completely go away, but it's severely reduced. So the eye becomes much darker. And so I photographed a dead animal next to a live one, next to a dark standard, and found that yes, there is a significant increase in contrast with the removal of eye shine. So it seems that eye shine is acting as a way to reduce contrast with the eye. But is it spectrally matched to the environment? And so what we did is, because these animals are so tiny, they're actually only about 2 millimeters long, we couldn't measure the light reflected off of an eye in the natural environment while we're on scuba. It's just not possible. We had to do that under a microscope, which was set up here, where we illuminated the eye at a 45-degree angle, and then imaged it through a microscope and measured the light spectrum coming off of the eye with a spectrometer. We also, while collecting the image data, I have my lovely assistant, Megan Porter, operating an underwater spectrometer, where she measured the downwelling radiance, so all of the light that was falling on the scene, as well as the radiance, or the spectrum of light, in every single direction that we measured And she did this at the exact same time that I was taking the photographs. So we know what the background light spectrum was at the time of the imaging. So if we then take the eye shine that we measured in the lab, and we multiply it times the downwelling irradiance, and we compare it to the sidewelling irradiance of a given condition, we see that we have an excellent match between the eye shine and that background light environment. So just as a check to see if this is really true, that this eye shine has evolved to match the background of the given environment the animal is in, we then did the same calculation, but with the Stomatopod species, which you recall is from the Western Atlantic Ocean, which, if you have any experience with the East Coast U.S., that water does not look anything like you would find in the Great Barrier Reef. It's much more long wavelength shifted with greener. And so if you take the eye shine reflectance from Stomatopod, you multiply it times the irradiance of a lizard island situation, and you compare it to the lizard island light spectrum, you get a horrible match, whereas the Poyoscola is a much tighter fit. And so that gave us some pretty strong support that eye shine is in fact used as a camouflage in the pelagic environment. And this is actually the first example of a reflective camouflage strategy in the pelagic environment that's matched to the background. You might have noticed that fish are silvery. That's a common reflective camouflage strategy for reducing contrast, and they just do it by wearing a mirror on their side. But they can do that because so we wondered in relation to this, especially since it's such a novel mechanism or a novel thing that we've uncovered, what are the structures that are responsible for producing eye shine? So I performed transmission electron microscopy in order to determine the ultrastructure of the eye shine material. And as I mentioned before, we predicted that the eye shine structure would be found between the optical layer and the retinal layer. And indeed, we found something that looked very photonic in nature when we looked at that. And it turns out that eye shine is produced by an amorphous solid, which is just it is a structure that is packed with vesicles that have a diameter on the scale of visible light, so between 400 and 700 nanometers, 307 nanometers in diameter, and that the geometric order of those vesicles, you see in these pictures from 4 different species, that order deteriorates the farther you get away from them. So you just have this sack of vesicles that are all packed together, and they are producing eye shine. Unexpectedly, in one species, Poyoscola thomasini, we found the amorphous solid that you can see up here, but then we also found this very unusual photonic structure within the photoreceptive unit, within the rhabdome, that has extremely long range orders. So that geometric order among the vesicles inside is preserved with distance. And these are just photographs of them within the rhabdome, where you have your optics up here, rhabdome, and then rhabdome on either side of the rhabdome. This is actually in an early stage animal, and this is actually in one of those larval retinas that's degenerating as the adult retinas form. So this is a very stable structure, whatever it is. The actual microvilli of the rhabdome are beginning to degenerate, but this stuff is still looking great. An interesting thing about Poyoscola thomasini that may be related to this structure is that it has an unusual eye shine. So if you look at the animal dorsally, it has this very bright green eye shine. Whereas if you flip the animal over on its back and look at the ventral aspect of the eye, it's very blue. We have this two-tone eye shine that we suspect may be some sort of counter shading with the camouflage function, but not quite sure. So these are just some zoomed in images of this insanely ordered photonic structure. It's made up of 4 different cells that come together and have these ordered vesicles that the order is actually preserved between the cells, so that's crazy. And this is a longitudinal section where you just have this very, very ordered array of structures within it. So possible functions are that perhaps it's contributing to at least maybe the green dorsal eye shine, looking at preliminary reflectance spectra that have been calculated from this structure. Or perhaps, since it's located within the photoreceptor, it's acting as a filter. So if you recall, all stomatopod larvae, at least that we've examined to date, only have a single class of photoreceptor in their eye at one time. So perhaps this is creating a dichromatic visual system by driving those photoreceptors that are beneath it away from the ones that are above in terms of their spectral sensitivity. So creating two spectral classes of photoreceptors. Why they would want to do this? That's yet to be determined. So future directions with this. Unfortunately, since we didn't expect to find this when I went to go do the TEM, we've never seen it in fresh tissue. So the logical thing to do is to go and look at and measure the interabdominal structure in fresh samples of tissue to measure the optical properties of it. I'd also really like to determine the 3D arrangement of the vesicles using techniques such as scanning SEM in order to determine how these vesicles are arranged in 3D space, so that we then can develop a more accurate model of how eye shine is working. So this is just a preliminary model that my collaborators at the University of Bristol have developed, demonstrating the histogram here shows the predicted reflectance from this particular structure compared to the measured eye shine reflectance. And you can see it's close, but it's not quite there in terms of ready for publication, so we need to develop that. And then I'd really like to test the eye shine spectral contrast theory in different spectral light environments, such as taking Squilla and Cusa out into the Western Atlantic and measuring its contrast and spectral matching. So with that, I'd like to summarize my talk. I demonstrated the use of DNA barcoding as a method for larval identification. I didn't really highlight that I used it as a retroactive method of identification. So I could, what I could do is I could go out and I could collect an animal, I could do an experiment on it, even if it's an experiment that terminated the animal, and then I could identify it later, which is very informative because conventional methods of animal identification require rearing larvae into adulthood, which if you're doing an experiment where you have to cut up an eyeball, you can't do that. I also demonstrated some of my results investigating larval photoreceptor sensitivity and diversity among sympatric species. A study that I did on the double retina light sensitivity and how ontogeny, or how sensitivity to light changes with ontogeny. I covered my series of experiments about larval eye shine, demonstrating that it is a contrast reducer that is spectrally matched and is made up of ordered vesicles. And with that, there are a lot of people to thank. So bear with me. Here it goes. So this is a team of researchers that I work with in the field at Lizard Island, taken at Lizard Island. Megan Porter has been a wonderful source of inspiration, help, mentorship. She was very involved in a lot of this work. Nick Roberts and his team of students, David Wilby, Tom Jordan, and James Foster, and Ilsa Bailey are all at the University of Bristol, and they've been wonderful colleagues in the field and for helping me develop the eye shine modeling. Also, my advisor, Tom, Dr. Jonathan Cohen was very helpful, or was really key to the electrophysiology experiments. He used his rig to train me to do that. Roy Caldwell is an expert in the field of stomatopods in general. He knows a lot about them and has always been very helpful in the field and in the study of larvae. So thank you, Roy, for everything. Alex Kingston, Richard Forward was very generous in letting us use his facilities at the Duke Marine Lab. He's my science grandfather since he trained Tom. Mike Bach also. What? Oh, and John, actually. Mike Bach, former lab mate. Also, thank you to Shane Awyong, who at the Australian Museum is one of the leading taxonomists on stomatopods and trained me to do my illustrations for morphological descriptions. Jenny Gum was very helpful with my statistics and using R and just generally being awesome, and a former roommate. Sonka Johnson was helpful with the eye-shine contrast. Thinking about it. Ellis Lowe graciously loaned us his field portable microspectrophotometer so that I could achieve my MSP study. And Short, former member of the lab, is very helpful and generally in getting me started on a lot of this work, as well as all of our funding agencies. Also like to thank my committee, past and present. Unfortunately, I had to do a little shakeup for this defense in order to meet the graduation timeline. But thank you, Phyllis, Jeff, Marjorie, Kevin, Tammy, and Al. You've all been very helpful over the years. It's been a wonderful experience. I've really loved graduate school. I'm not sure many people who've put this much time in could say that at the end, but this has been the best decision I've ever made. Everyone at UMBC and the biology department in general, just been wonderful. John Capaldi is a magician. He is our machinist, and basically you can come up with a vague concept of an apparatus, and he will take that idea and he will make it better and more beautiful than you could have possibly imagined. So he plucks it out of your brain and just casts a spell. Esther Fleischman, thank you, Esther, for being a wonderful mentor for teaching and showing me how to teach A&P. I feel like it's going to be my future. I love the subject and I love your teaching style, and I will carry what I've learned from you all the days of my life. Melody, Sue, Kristen, in the office, you guys are a joy. It's lovely to say hi to you, and all the things you've done for me over the years. You're just wonderful to have around. You do a great job. Cherie, who runs the imaging facility, has always been there to help me out when I have a need for doing TEM. Ralph, you're awesome. Thank you for everything. Latina is my current roommate for the past 2 years, and she is great. It's been such a joy living with her and being able to just hash things out at the end of the day with a fellow colleague, as well as the rest of the graduate students and alums from over the years. I also had the honor of taking the Loon Sensory Ecology course in Sweden this fall, and I learned so much about the field of sensory ecology, and it's really shaped me as a researcher in the directions that I really like to go. So thank you to all of those people. The iPods, which is the code name for everyone who works in the field. The Girl Science Posse, which is our 60 lady scientist crowd, as well as Kentaro Arakawa, who was graciously offered me a position in his lab beginning in January in Ayama, Japan. Ancillary to graduate school, I've had a very broad support network of non-scientists or science affiliates. So my mom and dad, thank you so much. I would not be here if not for your wonderful upbringing and support over the years. My sister Amelia, my cousin Meredith, they both heard I lived here in Maryland and then decided that they needed to live here too. So it's been really wonderful having actual family really close by. It's been wonderful. My grandma, while she never really understands what I do, has always been very supportive. My cousins, Kathy and Laura, my friends, Katie Bush, Lisa, Lizzie, Clark and Aaron, and Clark and Aaron Cricket, who are sitting here. They drove all the way from Western New York today, despite the snowpocalypse that you might have heard is happening. So thank you for surviving the journey. Thank you for being here. I love you guys so much. My Baltimore friends, Andy McKinley, Kurt, Anna, Karen, Anna and Scoogie, Rachel and Mark, Barbara and Adam, Michael and Chase, Paul and Andy, as well as my undergraduate advisor, Christy Kenyon, who really took a chance on me. And I wanted to do research. I had no idea really what it entailed. She said, I'm starting this project with bats and their eyes. And I said, yeah, I love bats. Eyes are great. And it really has just set me on a journey that I really have enjoyed very much. As well as my extended family and Creation Week Camp family is a camp that I look at and used to spend as a child, as well as so many others, and the love of my life is a guy named Larry, who, because I'm a deadbeat mom, lives in New York. And with that, some very special thank yous to my lab family. You guys are really, why this has been so wonderful. I'm not going to cry because it's going to make Alex uncomfortable. So Mary, I'm sorry, this is the only picture I have of you. It's terrible. But Mary also helped me with the ERG experiments last summer in the field, working around the clock. She was a real trooper. It was a really tough trip we went on. Alex, every day we do something, we say something. You are the glue that has been keeping this lab together and organized since Megan left 2 years ago. And you are a real rock in the lab and a joy to have around. Yes, beautiful. Mike Bach, who is one of my dearest friends and nemesis, all rolled into one. He is currently in Sweden, but while I have a running thing with watching him sleep and threatening to kill him, I really love him from the bottom of my heart. He's been a wonderful collaborator and colleague over the years. He's really been missed this past year since he's been in Sweden. Megan Porter, my former roommate of 4 years and inspiration for being visual ecologist in present day, and really helped me stay the course and smile when things got tough. But we've been on some great adventures. She's one of my dearest friends, and thank you for everything, Megan. And I'm glad you're able to tune in today via the YouTubes. Hope you can see these pictures. This doesn't seem to be great. And then Tom. Tom, thank you. Tom just took a very enthusiastic, kind of naive post-undergrad, and from the moment that he replied to my initial email, he was nothing but supportive about me joining his lab and going down this road. And he really has been the best mentor, better than John Cataldi could have even imagined. So thank you for turning me into the scientist I am today. And I look forward to our collaborations together. And generally, thank you to my lab family. I love you guys very much. I miss very much. Also, thank you to the new people in the lab, with Alice, John, and Tom, as well as Short. And above all, thank you to the hundreds of larvae that made this research possible. And thank you for coming.
I'm not sure how long we have this room. Does anybody actually know?
I think till 1:30.
Okay, then we're good.
Questions?
Other questions for Kate here?
Is there a larva in that picture?
No, this is an adult. This is a Haploscola trispinosa.
I thought there might be an invisible adult.
I mean, there might be larvae here. I've actually never seen them during the day, so they might be here in front of us. They're really good at hiding. The only way I'm able to capture them is actually to exploit their innate desire to swim for the light. Yes. Thank you. Yes. So some of the adults do have reflecting pigment in a similar region of the eye. Tends to be green or gold, especially in the Scolenophusa species. There is one ultrastructure study that I found that shows that it's not this ordered vesicle array, but rather a multi.
We got a
question. Let me finish answering this one. But it seems like it's a multi-layer reflector. But in short, no one's really characterized the ultrastructure of that immensely.
So I think we're ready for your question. Okay. It's Megan.
Hi, Megan.
Go ahead. Megan's asking, why do you think you could measure responsive to adult eye and adult eye, but not see anything done on the screen the same?
So there's a couple ideas with that. So in that, I mean, she's watching, so I can just talk to her. So there's a couple ideas with that. MSP is a very traumatic experiment to the eye, to say the least. It requires dissecting it off the animal and cutting a thin section of about 12 microns and then shining a small beam of light through the rhabdome that remains on your preparation. And so there could be a very small amount of visual pigment in the rhabdome that you're not able to measure because it's in such low quantities. So you're just using such a small amount of tissue. Also, there could be something, so in preparing the animal for microspectrophotometry, you have to keep it in the dark for hours to overnight. And so it's going through this critical developmental period that light might be required in order for the retina to develop normally. And so there might be something in that the visual pigment is not being ramped up in enough quantity to measure. MSP is a great technique, but not for measuring emerging adult eyes and double retinas. So.
You okay with that? Okay. Thanks very much for calling.
Thanks for your question, Megan. Yep.
For your UAGs, have you tried longer periods?
We have not. So that entire ERG study was conducted within an 8-day window, and that included driving down to North Carolina and capturing the animals and doing the ERG. So when I say it was a trying week for experimenting, I'm under-exaggerating. So we were pretty limited on the diversity of techniques and investigations we could do. So that would certainly be an interesting thing to do, is to test the duration. But that explains why we didn't do much more than that.
You know where ERG
it should be. It's measuring a change in the potential of the actual photoreceptor cells. So it's, I mean, the tip of the microelectrode is right in there. It's possible it could be coming from the lamina, which is the neural pal just beneath the retina, but it's actually the retinular cells. They're as developed as the retina is, since they're actually, the lamina is formed from the projections from the retina. So that's why I'd like to do the neuroanatomy characterization, figure out what's going on there. Does that answer your question?
If you guys don't have any more questions for Kate, her committee has met. So this is your last chance to ask her.
Thank you.
How was that you had your calls on?
And
What is
thank
that
you to the
thing you
YouTube people.